Enzymes. These little shits do literally everything in your body. You love them, you hate them, you gotta know them. That's not the phrase. Whether you hate them or you love. Okay, I'm not even gonna try knocking that again. But, anyways, today we're gonna be talking about the different types of enzymes. And I'm gonna break what they do down for you with examples on what actually happens in your body so that you're able to make a connection a little bit easier. I think it's incredibly important whenever you're studying biochemistry or just biology in general to realize that everything you learned is meant to be interconnected with one another, right? So just simply brute force memorizing what each enzyme does, or just being like, oh, this catalyzes that, that catalyzes that, like you're memorizing some fucking SAT vocab, is not going to serve you. What is going to help you is to make connections to concepts that you've learned before. So, okay, I understand what a hydrolase is, but where in my body does hydrolase work? It's intuitive to think that, oh, the digestive system, because hydrolase is going to use water to cleave shit, and your digestive system, the whole point of your digestive system is able to cleave big shit to make little shit that can be absorbed into your body, right? So hydrolases are probably going to work in your body there. Like, those are the types of concepts and connections that I really want us to start fostering. That sounded incredibly formal. It's not that deep. We're just going to do it, okay? All right, so I have this graphic that I'm working with you guys today. And then, okay, you guys get to choose. Where do we want to start? Let's actually start with the most intuitive part. We'll go clockwise, okay? All right, so enzymes. At this point in time, when you click on this video, I really hope you guys do know what an enzyme is. Basically, it's a biological catalyst that doesn't change anything about the reaction except its rate. Now, what does that mean? So imagine you're going to the gym, right? And then you hit a workout without taking pre-workout. You might not perform as well, right? You might not be able to lift as much weight, or maybe you're not running as fast as you could. Whereas if you take pre-workout, maybe you're able to do one more rep. Maybe you're able to run a little bit quicker. But does taking pre-workout change who you are? No, right? Does it change like your muscle composition immediately? No. Enzymes are the same thing. Adding an enzyme isn't going to change the nature of the reaction in the sense that it's not going to chemically alter any substances. It's not going to chemically alter how the reaction proceeds. What it will change is the either likelihood that the reaction happens, and then what it will change is the rate as which the reaction happens, right? That's why it's called a biological catalyst. Catalysts speed things up, right? With that out of the way, we're first going to look at isomerases. So quick note, whenever you're looking at enzyme names, right, you'll always notice that enzymes will always end in ASC. And that's going to be almost always, I want to say with 95% accuracy, true. An enzyme, if you see ASC at the end of a molecule name, chances are it's going to be an enzyme. Whenever you're talking about enzyme functions, always look at the name and kind of try to figure out what they're going to do, because most of the time enzymes are just going to be named the way that they do shit, right? So isomerase, intuitively, what do you think an isomerase is going to do? It's going to catalyze the interconversion of isomers, right? What are isomers? Molecules that have the same chemical formula but just differ structurally. So these two would be isomers because it's literally the same except the position of this little blue thing, right? Isomerases are going to be the things that actually catalyze this reaction to go quicker. A noteworthy type of isomerase is going to be mutase. So mutase, again, name is kind of intuitive. It sounds like it's going to mutate something about the molecule, right? And you'll be right. Remember glycolysis, our phosphoglycerate mutase. What did that do in glycolysis? It catalyzed the interconversion of glycerate 3-phosphate to glycerate 2-phosphate, right? So yeah, mutase is going to be a type of isomerase because those two molecules are isomers, and mutase is going to be the one that does this. Really intuitive, really easy to remember, right? Oxidoreductase. Again, look to name to think of function. What do you think intuitively oxidoreductase is going to do? This is going to be an enzyme that catalyzes redox reactions. So that's going to be your oxidations, and that's going to be your reductions. So interestingly enough, there are two main categories of this enzyme. It's going to be your oxidase, which catalyzes oxidation reactions, and your reductases, which catalyzes reduction reactions. So that's kind of intuitive. What I do want you guys to note is actually the existence of dehydrogenase. as a type of oxidoreductase within this category, right? And where have you seen dehydrogenase? The citric acid cycle, right? The whole point of the citric acid cycle is to oxidize the members that are involved within the cycle, right? And then an oxidation is what? Oil rig, remember oil rig. So oxidation is the loss of electrons, while reduction is the gain of electrons. So if a substrate within the citric acid cycle is oxidized, that means it lost electrons to an electron carrier, which goes to the ETC, and that's the whole point of the citric acid cycle, right? But I digress. Dehydrogenase was a very potent type of enzymes that was in the citric acid cycle. We have isocitrate dehydrogenase, which what? Dehydrogenated isocitrate, i.e. it oxidized an isocitrate. Every time you take away a hydrogen, i.e. dehydrogenase something, that means the molecule has been oxidized, right? So I want you to really remember dehydrogenase as a type of oxidoreductase, especially within the citric acid cycle. All right, so we're gonna go to what I call the Ly sisters. So we have ligase and we have lyase. I know, they sound so similar and it's so annoying, but they do very, very different things. They actually do opposite things. So ligase, where have you heard ligase before? Do we kind of remember DNA ligases? I hope we do. This particular class of enzymes play a very significant role in DNA replication and repair. So that whole process is going to use a lot of ligases. More specifically, some noteworthy enzymes within this class is synthetase, which does what? It adds amino groups to tRNA to be able to synthesize it. So we have our tRNA, and then we have our growing chain of amino acids within the translation process, right? Now this process, this building of molecules, is going to be catalyzed by a synthetase, which is a type of ligase. So ligases always build. How I like to kind of remember it is that the G in ligase stands... I just got a southern accent there randomly, my bad. What the fuck? Anyways, I like to think that the G stands for gain, right? So you're gaining some sort of mass, I guess, in a sense. You're gaining bigger biomolecules. That's what ligases do. Another way you can memorize it is that G stands for glue, right? So, ligases, G stands for glue. So ligases are going to glue molecules together, and that's exactly what DNA ligase does. So if you remember, during replication, we're going to have Okazaki fragments in the lagging strand, right? So those are going to be fragments of DNA that is continually synthesized. And in order for us to have a continuous chain of DNA, we're going to have to glue them together somehow, right? So basically, it's going to catalyze this reaction, where now it's one bigger biomolecule. That's what ligases do. Ligases glue. Lyases. So it's like ligase's evil twin. If ligases are out there building shit for us, lyases are like, fuck you, I'm gonna break these down. More often than not, I feel like people get confused with lyases because you see so many other enzymes where like, oh, hydrolases, like hydrolysis, like I guess that's just gonna break molecules together. So what the hell is a lyase, right? And then you also probably saw in your textbooks or wherever that lyases break and make double bonds. And then intuitively, that just sounds weird. Like, what do you mean, right? So in order to understand lyases really well, we look to the citric acid cycle again, okay? Remember pyruvate decarboxylase? Now, again, look to enzyme name, pyruvate decarboxylase. So obviously, this is going to be an enzyme that takes a carboxyl group or a CO2 group more easily, a carboxylic acid group or carboxyl group out of pyruvate, right? So if you look to the pyruvate molecule here, this is a lyase, and then you'll see that a CO2 molecule left. Now, where did that CO2 come from? It came from breaking this bond. So lyases, so this is my enzyme. Lyases are going to lyse this bond, and it's going to release CO2. Now, notice that CO2 leaves with a double bond here, right? Because CO2 has two double bonds to an oxygen. So that's what textbooks mean when they say lyases create double bonds when they break bonds, because it just kind of intuitively doesn't make sense until you actually see it in action. So CO2 left with another double bond on its oxygen. So this bond has been lysed. Another example of lyase is going to be aldolase. So aldolase isn't in the CAC, my bad. The aldolase is in glycolysis, right? I obviously have a thing for glycolysis. Now, don't freak out. This is fructose 1,6-bisphosphate. And I know you're more used to looking at this molecule in its cyclic form, right? So we have a phosphate group here. We have a phosphate group here. You can rarely do anything in the cyclical form, right? So if you open this up, this is what fructose 1,6-bisphosphate is going to look like. But I'm not too concerned about you knowing what that looks like. I'm more concerned about you seeing where the bonds broke. So this is fructose 1,6-bisphosphate, right? And that's our DAP, and that's our GAP molecule. Remember, GAP molecule is going to proceed to glycolysis, right? But anyways, more importantly, aldolase is going to be the one that breaks the bond right in the middle of this open chain form of fructose. So it's easier to visualize once you see how the two molecules are structured, right? Do you see how these two, DAP and this half of fructose 1,6-phosphate are the same, and then GAP and this half is the same? Now, where do you see the double bond that we talked about earlier form? It's right here, right? In glyceraldehyde, in the aldehyde group of glyceraldehyde 3-phosphate, that aldehyde group, the double bond formed right here when this bond broke. So if it makes it easier for you to visualize, you can also visualize this bond that's being broken being moved here. So if it helps you visualize a little bit better, whenever you're working with a lyase to see like what the enzyme is actually doing, take the bond that's being cleaved and just imagine it going into a double bond, right? And that's basically what lyases do. And then I know that lyases are a little bit less intuitive in terms of their names, but if it becomes easier if you think about lyases lysing. So if ligases glue, lyases are gonna lyse. And what does lyse mean? Just breaking apart, right? This is kind of an unhinged side note, but when I first started learning what lyases do, I would just kind of like make it fun for myself and be like, wait, you're like fucking lying. So you're telling me that this enzyme can literally like break bonds without even using water or the transfer of electrons? Get out of here. She's doing too much. Like, that's how I memorized it. I never feel like more of a pathetic ass little nerd when I'm making up these like mnemonics or like just like memorization devices for myself. But sometimes you just got to do what you got to do. So lyases, you're fucking lying. It's able to do what every enzyme wants to do without the help of water or electron transfers. Insane. But anyways, or if you're normal, just memorize lyases, lyse, ligases, glue. Okay? Anyways, moving on. Hydrolase. So hydrolase should be really intuitive to understand what they do. Hydro, water, lase, right? Hydrolysis is probably going to do something with hydrolysis, and you will be right. Hydrolases basically take a molecule and with water breaks it in to its component parts. Now, I talked about this briefly earlier, but where do you think hydrolyses are going to work the most? It's in the digestive system, right? Because the whole point of the digestive tract is to lyse bigger molecules into smaller molecules so that they're able to be absorbed by the intestinal tracts, right? It's also like kind of intuitive to remember that like water is in the digestive system, because I don't know about you, but whenever I imagine the digestive system, it's just really wet and gross and slimy in there, and that's how I like to kind of like conceptualize it. But anyways, this is also the primary reason why you feel bloated when you're dehydrated, because digestion really requires a lot of water to be able to work really efficiently and well, because hydrolase requires water to work well, right? So the next time your man asks you if you drink water when you tell him that your life sucks, I don't know, still be mad at him because that's annoying, but like maybe drink a glass of water and then maybe you'll hate him a little bit less, you know? Anyways, all right. So the three types of hydrolase is really easy to remember. It's going to take our three macromolecules, like our three biomacromolecules, and it's going to break those down. So lipases, really intuitive. Lipase, LIP, lipids. So lipases are going to break lipids down into its component parts, again, using water. Remember, all of these are always going to use water because all of these are types of hydrolases, okay? So using water, lipases are going to break lipids down. Proteases, what do you think that's going to do? Sherlock Holmes, proteins, right? It's going to break proteins, i.e. the amino acid chains, into its component parts. Finally, nucleases, whatever the fuck, is going to use water to break down large nucleic acids into its component parts as well, right? Again, using water because it's a type of hydrolase. Our final group is our transferase. Again, this should be really intuitive. Transferases are probably going to transfer something, right? And you'd be right. That's exactly what they do. Transferases basically transfer groups of molecules from one molecule to another. There are three very, very important types of transferases that you absolutely need to know. The first one being your old friend kinases. So kinases are basically going to phosphorylate a group using the transfer of a phosphoryl group from an ATP specifically. So I drew an ATP out here for you. The yellow groups are going to, represent your phosphate groups, right? And then this is your kinase. So kinases, like you can have pKa, pKb, etc., pKc. I don't know, they just keep coming. Or like you can have pyruvate kinase. Anytime you see a kinase, always think ATP and a transfer of phosphate groups. Now, phosphorylases do a very similar thing, and this is where a lot of people get tripped up on like, okay, what the hell is the difference between a kinase and a phosphorylase? So there are very niche difference between them, but we're not going to go into that today. Basically, what I want you to know is that phosphorylases do, Phosphorylases do the exact same thing that kinases do, except not with an ATP, but with just like a organic phosphate group. So it's literally just going to take a PI group, a phosphate group, and attach it. Same exact thing as a kinase. Is just going to use a phosphate group in itself instead of using ATP specifically. All right, we reached our final type of enzyme, which is a type of transferase, and that's going to be phosphatases. So we better have something that actually is able to take this off, right? So phosphatases are going to help out the reverse reaction of this, right? So if we have a molecule that's phosphorylated, right, phosphatases are basically going to take away the phosphate group. That's how I want you to like visualize phosphatases. It's gonna take a phosphate group away, while phosphorylases are gonna give phosphate groups to the molecule in question. That's an overview on the bare bone essential types of molecules and enzymes that you want to know. Let me know if you guys want any like specific types of videos or specific videos on specific concepts. I'll try my best to dumb it down for both of us, actually, because heaven knows I need it too.